This next section is on viral replication. And I know that we previewed this in chapter 9 when we talked about transduction. So we had to do a little preview of the lytic, or as some people call it, the lytic cycle, and the lysogenic cycle. So we've already had a little preview of that. So if you take a look at this picture, we actually have the lytic cycle on one side and we have the lysogenic cycle on the other side. So if you remember with the lytic cycle, we had that virulent phage. So there's our little bacteriophage, and that is going to infect a bacterial cell. So it has to first attach to that cell. If it can't attach, all bets are off. It's not going to be able to replicate. If you remember, one of the terms that I mentioned for attaching is called adsorption. So that bacteriophage adsorbs, it docks onto that cell, injects its DNA. And remember, this is the violent quick cycle with that virulent phage. And you can see where that DNA comes in, takes over the host cell machinery, makes its parts, and then assembles those bacteriophages. And then you can see that last picture where it's lysing the cell. Now all of those bacteriophages can go out and they can infect other cells. On the other side, we've got the lysogenic cycle. That's the longer, sneakier cycle. That's that temperate phage. So again, that bacteriophage, it has to dock, it has to adsorb. So this is one thing that the lytic and lysogenic cycle has in common. They both have to be able to adsorb to the cell, inject the DNA into the cell. That's another thing that they both have in common. But remember, it's not going to take over that host cell machinery right away. It's going to integrate into the host DNA, and then we call it a prophage. So with that middle picture, you can see the green bacterial DNA and the red bacteriophage DNA, which is sneaking into that DNA. Now, the cell has no idea that that viral DNA is integrated, and it just keeps dividing over many generations until something happens. Maybe the cell is stressed out, maybe it's been exposed to chemicals or radiation, and that DNA, that viral DNA is like, I'm out of here because this is going south fast. And now we're going to cross over into the lytic cycle. This crossing over and going into the lytic cycle is what we call induction. So induction is going from the lysogenic to the lytic cycle. Now that viral DNA is going to take over the host cell machinery, make its parts, and then eventually lyse that cell. So I tried to pick a very simplified picture just as a little side by side, because coming up, we're going to look at a little more detailed picture. So if we were to write out each of these phases of that bacteriophage replication, first we have adsorption. That's that attachment to the host cell. If that virus comes in like a balloon and kind of bounces off, all bets are off. It's not going to be able to replicate. The second thing is that it's going to have to inject the DNA into the cell. This is what we call penetration. So if you notice that previous picture, in both the lytic and lysogenic cycle, we were seeing adsorption and we were seeing penetration. Then things sort of diverged, where one was hanging out as a prophage, the other one was taking over that host cell machinery right away. So when it takes over that host cell machinery to make its parts, so this is going through transcription and translation and replicating the nucleic acid, this is what we call synthesis. You'll notice in one of the upcoming pictures, they also call this biosynthesis. Either term is fine, either synthesis or biosynthesis. After that happens, now we've got all of those parts. We've got the nucleic acid, we've got the protein coat. Now we have to put them together to make those new virions. This is what we call maturation. So this is putting all of those parts together Now we have these new virions. Now, what are they going to do? They're not going to continue to do anything else in the cell. They need to go out and find another cell to continue with that replication. So that's when we have release. So the cell blows open, it lyses, and now all of those new virions go out and escape, and now they can infect other cells. So if we're putting this in word form, adsorption, penetration, synthesis, maturation, and release. Now, if you like mnemonics, this would be a good time just to kind of take each of those first letters and maybe think of a mnemonic that maybe you can relate to. Believe it or not, as much as I love letters and mnemonics, I actually don't have one for this. This is one where I think more visually and think about what's actually happening with that viral replication. So if you're like me and you are visual and you like to see a picture, here's a picture of what's happening. Now, if you take a look at this picture, take a guess at what stage you think this is. Looking at this entire picture, this is the lytic cycle, right? So if we start over on the right-hand side, you're going to see number 1, adsorption. Bacteriophage has to attach. It's got to dock to that cell. Second phase is going to be penetration. So you can see that it's injecting that DNA into the cell. Now that bacterial cell is disrupted, it's going to take over the host cell machinery. This is why we call viruses intracellular obligate parasites. They cannot replicate unless they take over that host cell machinery. So on the bottom, you can see 3 is calling it biosynthesis. Either term is fine. And now it's taking over that host cell machinery. Now remember, that host cell machinery has to be compatible. So the first part is that virus has to dock. It's got to adsorb. Then all of the machinery, the enzymes have to be compatible to make all of its parts. So in synthesis phase, this is when the nucleic acid is replicated, proteins are made. Maturation, now we're actually going to assemble these bacteriophages. And then finally, that top picture, you can see the release. Now, the cell is a goner because these viruses are going to blow open, lyse the cell, and now they're going to go out and they're going to infect other cells. So this picture that we're looking at is actually of the lytic cycle. Now, putting both of these together, we actually have the lysogenic and the lytic cycle. So if we take a look at the middle of the picture, and I'm going to get my pen just to kind of point this out. Here we've got our cell, right? So we've got adsorption and penetration. Bacteriophage adsorbs, docks on the cell, and that DNA penetrates into the cell. So remember, this is something that both the lytic and lysogenic cycle have in common. So let's follow the bottom picture because this is going to be lysogeny. So DNA enters. It's not going to take over right away. It's actually going to exist as a prophage. So if you notice in this middle picture, we've got sort of that purple bacterial DNA and the red bacteriophage DNA. That bacteriophage DNA is just going to hang out there. The cell has absolutely no idea that that DNA is there, so it's just kind of going about its business. So it's going to be replicating its DNA and dividing. When it replicates its DNA, it's also going to replicate that phage DNA. And then we're going to have many generations of the cell. So here with this picture, you can see where we've got binary fission happening. So we can go through many, many cell divisions. All right, so perhaps something happens in the environment. Maybe there's not a lot of nutrients. Maybe the cell is stressed out. Maybe it's been exposed to chemicals or radiation. The cell is not doing so hot, right? So now that viral DNA is like, yeah, I think I'm kind of out of here now. So now we've got induction. So here's induction. What is induction? That's when it goes from the lysogenic cycle to the lytic cycle. So now we're going to cross over, and now we're going to take over that host cell machinery, go through synthesis, make all of the parts. Here we've got all of the parts in this picture. Maturation is assembling all of those bacteriophages. And then finally, we've got the release. So now you see the release of these bacteriophages. These bacteriophages can go out and they can infect other cells. So this is taking a look at the lysogenic cycle and then crossing over into the lytic cycle. This picture right here is how an animal virus would replicate. Very beginning, we kind of went over the generic structure of a virus, and we mentioned the envelope. So remember that envelope. So it's that lipid bilayer. Where did that envelope come from? It was stolen from the host cell as it was escaping. So if you take a look at this picture right here, one of the things that we said about the envelope that was an advantage is that it actually allows that virus to gain access into the cell. So if you take a look right here, as we kind of meet up with the membrane of that cell, that virus with that envelope can kind of meld into that cell. So it kind of gives it an easier passageway into that cell. Now it's going to take over the host cell machinery. You can see this is a eukaryotic cell because we can see our nucleus there. So what's happening in the nucleus? Well, we're replicating the nucleic acid, going through transcription, going through translation, and now assembling those new virions. Now it's going to be time for that virus to actually leave. Notice its exit point right here on the bottom. As it exits, so over here in the nucleus, see how it's naked? It doesn't have an envelope yet. Now as it leaves, it's wrapping itself in this envelope, almost like this cloak that it's wrapping in. So it's stealing that membrane, and now we have that virus with that envelope. So it has stolen part of that membrane. Remember, if a virus cannot attach to a cell, then all bets are off. It's not going to be able to do anything to that cell. So like I said before, if it kind of comes in like a balloon or a ball and kind of bounces off, that cell is not going to be infected. So that adsorption, that attachment is what is going to be critical to that virus replicating. So this picture right here is looking at those attachment sites. So this picture A that we have here on the left-hand side, you can see that this virus does not have an envelope. So it has that exposed capsid. And remember that capsid is made up of those capsomeres. So here's the capsomeres. You notice that these capsomeres have these little divots in it, almost like a little puzzle piece. So what it attaches to, if you look at right here, is our host plasma membrane. So imagine this is an entire cell. On that host plasma membrane, there is actually a receptor protein. That receptor protein has a certain shape that actually fits in the canyon of that capsomere. That is allowing that virus to lock in and adsorb to the cell. If it did not have that capsomere that fit perfectly, like a puzzle piece fit, with that receptor protein on the host membrane, then it's not going to be able to adsorb and it's not going to be able to replicate. So here with our virus that does not have an envelope, it's going to use those capsomeres to adsorb. On the right-hand side, on this B picture, here we have a virus with an envelope. So remember those spike proteins, and this is something that they talk quite a bit about with developing the COVID vaccines, was that spike protein. And introducing that spike protein to the immune system allowed the immune system to recognize it as being foreign. Well, viruses with envelopes, they've got those spike proteins. And so that spike protein now is going to have a perfect puzzle piece fit with that receptor protein, which is on that host cell. So, the very first step, whether we're talking about animal viruses, whether we're talking about bacteriophages, even plant viruses, has to be that adsorption. Viruses have to attach to cells. Now, how is it that we can learn about these attachment sites like the spike protein? How can we learn about how viruses replicate and how they attack cells so that maybe we can prevent viral outbreaks and develop some treatments? The best way to really study viruses is in the lab in a cell culture. Now, we can have primary cell cultures that come directly from an animal. So we could take some cells directly from an animal and do some tests. So, how is that virus adsorbing to that cell? Problem with primary cell cultures is that they only last a short period of time. So, they're really difficult to maintain in the lab. There are some other types of cell cultures, but the most common type of cell culture that we see in lab are continuous cell lines. Continuous cell lines are cancer cells, and cancer cells are rogue cells. And we'll talk about cancer at the end of this chapter. Cancer cells do not behave like other cells, and they are basically immortal. They don't go through the normal checkpoints that a cell would, and so they just keep dividing and dividing. So there are many different types of continuous cell lines. I mentioned when I first started in pharmaceuticals, I had to get the Hep B vaccine because we were working with a cell line. It happened to be a cancer cell line. And so they recommended that anybody that was working with that cell line, get the appropriate vaccinations. The most famous cell line are actually HeLa cells. And there was a time that we actually used to stain HeLa cells in Bio 109, but they got very difficult to get and very expensive that unfortunately we had to drop that lab. But I do try to talk about HeLa cells whenever I can. And HeLa cells were actually derived from cervical cancer from a woman, and her name was Henrietta Lacks. And Henrietta Lacks actually died in the early 50s. She was very young. She was probably about 31, 32. And these cells get their name from her name. So HeLa cells, the HE is actually the first part, Henrietta. The LA is actually from Lax. These are an immortal cell line. There is a great book out there. It's called The Immortal Life of Henrietta Lacks. And it is a great book that talks about not only the cell line and these cancer cells, the amount of research that has been derived from these cells. We would not have the medicines, the vaccines without HeLa cells. And the amazing thing is that the woman that provided these cells actually had no idea. And she had no idea the impact that her cells would have on modern medicine. So Henrietta Lacks, she was a very young woman, around 30. She noticed that something was wrong. So she was in the Virginia area and she went to Johns Hopkins that was doing some work for people in the community. She was a very poor African American woman. And she went to Johns Hopkins to have this lump checked out. Turns out she had cervical cancer. So they ended up taking a little excision of her tumor and sent it down to a lab for testing. At the time, there was a lab that was run by Dr. George Guy, who was doing some work on continuous cell lines. And so when these cells actually hit his lab, these were the first cells that he was able to grow in culture, and they became an immortal cell line. He was very excited about this, and so he actually sent these cells to labs all over the world. And this is actually the most used cell line in history. They were so prolific that at one time they thought that all of the continuous cell lines were actually HeLa cells. You cannot imagine the amount of cells that have actually been used for medicines, vaccines. The book, The Immortal Life of Henrietta Lacks, which is written by Rebecca Skloot, she interviewed the family, and the family story is very compelling. They had no idea the impact that their mother's cells had until some researchers kind of showed up on their door to take some blood samples to find out, oh, maybe the families have the same type of markers. And so they started to learn about their mother's contribution. And so the story of the family is very compelling because they were not able to afford good medical health. It's an excellent book that goes not only into the science, but the compelling story of the family and the law as well. Many times in biotechnology, the law is kind of dragging behind, and it takes a long time for it to actually catch up. And it's one of those amazing stories, and I always say that we should give credit where credit is due. And so that's why I often talk about Henrietta Lacks. So I will post a video so that you can actually see a little bit about Henrietta and the family. But what is it about Henrietta's cells that allowed them to continue to divide in culture? Well, this is a picture of our chromosomes. And at the end of our chromosomes, we actually have these repeating nucleotide sequences. These are called telomeres. And telomeres, I often compare that to the little plastic covering that's on the end of the shoelaces. I believe that's called an aglet. And so that protects the end of the shoelaces from unraveling. Well, over time, if you've ever used shoelaces for a long period of time, that plastic starts to kind of fall apart and it gets shorter. And then the shoelace can actually start to unravel. Well, as our cells divide, there's actually a finite number of times that our cells can divide. And these telomeres kind of play into these checkpoints. So, as our DNA replicates, and if you remember learning about DNA replication, we talked about that leading strand and lagging strand. Because of that lagging strand that's made in pieces, every time our cells divide, these telomeres actually get shorter. So, after many divisions, these telomeres basically become non existent. This is actually a checkpoint for the cell to say, all right, we could be getting into some valuable genetic material that we don't want to lose. And so, it's a signal to the cell that, okay, we're done dividing. So cells actually have a finite number of times they can divide. We often call this Hayflick's number because Dr. Hayflick discovered that cells kind of had an expiration date. Well, the interesting thing about Henrietta cells and cancer cells is that they're rogue cells. They're not going to play by the same rules. They're not going to play by those same checkpoints. So, the interesting thing about Henrietta cells is that she had an enzyme that actually rebuilt the telomeres. So they never got old. They just kept dividing over and over again. And so this is what made Henrietta cells so unique and why we were able to use those cells to develop medicines and vaccines that we would not otherwise have. This is one of those things that I always tell students, if we could figure out why her cells had that enzyme, and if we could do that to other cells, we could figure out the fountain of youth. They've done some studies with some astronauts in space to actually look at the telomeres, and they found that astronauts in space, those telomeres actually get shorter, which is interesting. One of those studies. So if we could tap into this all as a group, we could actually be billionaires if we could discover the fountain of youth. But we did gain a lot of medical technology from Henrietta cells, which is why I always like to discuss Henrietta and her contributions.